Well, welcome everyone to today's session. We're very happy to have you join us. My name is Marie Latalipe and I manage some of the nutrition programs at ILSI North America. And today we have two excellent speakers and we hope to provide you with some tools to enhance your practice and maybe respond to questions from your patients or clients about sweeteners and the gut microbiome. And as people continue to join, we just wanted to give you some information about ILSI North America, which is the organization that's bringing you this webinar today. Very briefly, we are a nonprofit scientific organization that focuses on advancing nutrition and food safety science for the benefit of the health of the public. We always like to clarify that we do not lobby and we don't advocate for specific policies. We do our work through a tripartite model, and you can see that symbol up there to the right, and in which we bring together scientists from industry, academia, and government to address scientific questions that are of common interest. And among 12 area-specific committees that we have, one is the Low-Calorie Sweeteners Committee, which has the overarching mission that you see here. And actually, the committee organized a webinar last year focusing on sweeteners and safety with representatives from the FDA, Diabetes Canada, and AND, and that's actually available on our website for a credit if you would like to look for that. And we learned from that webinar that this area was of key interest to practitioners learning more about sweeteners and the gut microbiome. So this webinar is approved for one continuing education credit through CDR, and to receive that, you just need to respond to a very brief survey that you'll receive after the webinar is over. And one minor housekeeping point, I think we're all pretty used to this, but if you have questions throughout the webinar, feel free to type them into the chat box, and we'll bring those to the panelists at the end. And with that, just a final thank you to everybody for joining us. Please connect with us through any of the platforms that you see on the slide here. And I think we are ready to kick off the webinar. So I would like to introduce to you Dr. Cindy Davis, our first speaker. Dr. Davis is the Director of Grants and Extramural Activities in the Office of Dietary Supplements of NIH. And in this position, she actively engages and encourages partnerships with other NIH institutes and centers. She's also very actively involved in several government working groups that focus on the microbiome. So Dr. Davis will set the stage with an overview of our current level of scientific understanding related to diet and gut microbiome. So with that, Dr. Davis, we will turn the platform over to you.
Well, thank you, Marie, for that introduction, and it's a pleasure to be here today to give a bit of an overview about the relationship between diet, microbiome, and health. What I'd like to do today is give a little bit of background in terms of what is the microbiome, because I realize that you're all coming here from different levels of understanding. I'd like to give some of the evidence to suggest that diet can influence the microbiome. And conversely, how can the microbiome influence the response to dietary components? Interwoven throughout all of this will be what is the relationship between diet, the microbiome, and disease risk. So humans have been considered superorganisms. That's because we're a composite of many different species. In addition to our human or mammalian or eukaryotic cells, there are also bacterial cells, archaeal cells, fungal cells, and in fact, the estimates are anywhere from equal numbers to up to 10 times more microbial cells than human cells. When we talk about the microbiota, we're talking about all the microbes in a specific niche within the body. So the gut microbiota is all the microbes in our gastrointestinal tract. And in fact, this is about 100 trillion organisms. The microbiome is their collective genome. And in fact, there's about 100 times as many genes as the human genome. Other terms that you might hear is metagenome, which is a combination of both the microbial genes and the human genes, or metagenomics, which is actually the analysis of the metagenome. Dysbiosis is another popular term, and that refers to a dysregulated or abnormal microbiota. This could be abnormal in terms of the specific microbes that are present, or it can be in terms of the function of those microbiota. A dysregulated microbiota is common to many diseases, including obesity, diabetes, colon cancer, and inflammatory bowel disease, and all of those are associated with a decreased diversity of the microbiome. So what do microbes do for us? Well, they have a number of different functions. They provide the ability to harvest nutrients. They produce additional energy otherwise inaccessible to the host, and this is mainly from the fermentation of soluble fiber to the short-chain fatty acids. It produces vitamins such as folate, biotin, and vitamin K. It metabolizes carcinogens such as the heterocyclic amines formed in food during cooking. It prevents colonization by pathogens, and it assists in the development of a mature immune system. So what is the evidence that suggests that diet can influence the gut microbiome profile? In fact, there is a lot of different evidence. This includes such things as geographic correlation studies that have noticed that different dietary intake results in differences in gut bacteria in globally distinct populations. This was one of the first studies published about a decade ago that compared the microbiome profile in children living in Burkina Faso, which is a rural area in Africa, versus Italy. And the African children's microbial composition is shown on the top, and the Italian children's microbial profile is shown on the bottom. As you can see, the top profile has significantly more of the green color, which represents the Bacteroidetes. In contrast, the bottom panel has significantly more red and less green, and the red represents the Firmicutes. Why this is important is the specific Bacteroidetes are Prevotella and Xylanobacteria. And why this is important is these bacteria have specific enzymes that allow them to actually metabolize xylan and cellulose, so that these children can maximize the use of the carbohydrate of the dietary fiber and maximize the metabolism of this ingredients to get the most energy they can from their food source. Immigration can also influence the gut microbiome. In this study, investigators collected stool, dietary recalls, and anthropometrics from 514 Hmong and Karen individuals living in Thailand and the United States, including first and second generation immigrants, and 19 Karen individuals sampled before and after immigration, as well as from 36 US or European American individuals. Using 16S and deep shotgun metagenomic DNA sequencing, they found that migration from a non-Western country to the United States is associated with immediate loss of gut microbiome diversity and function, in which US associated strains and functions displace native strains and functions. US immigrants lost the bacterial enzymes associated with plant fiber degradation. And in fact, Bacteroides strains displaced Prevotella strains according to time spent in the United States. Finally, loss of diversity increases with obesity and is compounded across generations. So how important is diet in influencing the microbiome? An important paper published 6 years ago demonstrated that short-term consumption of diets composed entirely of animal or plant products alters microbial community structure and overwhelms inter-individual differences in microbial gene expression. I know this slide is fairly complicated, but I'll quickly walk it through you. All of the 10 subjects were on both diets for 4 days. The green represents when they were on the plant-based diets, and the red indicates when they were on the animal-based diets. As you can see on the top, when they were on the different diets, there were different short-chain fatty acids and other metabolites produced. Even more interesting is the bottom panel, which just looks at each of their stool samples and the similarities and differences between those samples. As you can see, the red tend to clump together and the green tend to clump together. And what this means is that diet is actually more important than genetics in influencing the microbiota composition. This idea that diet is more important than genetics in shaping the human gut microbiota was recently confirmed. Genotype and microbiome data were collected from 1,046 healthy individuals in Israel with several distinct ancestral origins, including Ashkenazi, North African, Middle Eastern, Sephardi, Yemenite, and mixed or other backgrounds. But a relatively common environment was compared. While genetic principal components were strongly associated with self-reported ancestry, there was no significant association between ancestry and microbiome composition. In addition, over 20% of the inter-individual microbiome variability is associated with factors related to diet, drugs, and anthropometric measurements. In contrast, host SNPs could not be used to infer a statistically significant fraction of beta diversity variants. And if you actually look at the top contributors, you can see that number 2 is vegetable intake and number 3 is meat intake. These results again suggest that environment has a substantially greater role than host genetics in influencing the composition of the human gut microbiome. Although diet is a key determinant of human gut microbiome variation, the fine-scale relationships between daily food choices and human gut microbiome composition remain unexplored. Multivariate methods were used to integrate 24-hour records and fecal metagenomes from 34 healthy subjects collected daily over 17 days. The investigators found that microbiome composition depended on at least 2 days of dietary history. Daily microbiome composition was more strongly associated with food choices than with nutrient profile. Similar foods have different effects on different people's microbiome. These results suggest that food-based interventions seeking to modulate the gut microbiota may need to be tailored to the individual microbiome, since not everybody responded to the same. So if diet is important in modulating the gut microbiota, what are some of the dietary factors that can be used to influence microbiota composition? The first is probiotics. Probiotics are foods or dietary supplements that contain live bacteria that, when administered in an adequate amount, exert a health benefit on the host. Prebiotics are non-digestible food ingredients which selectively stimulate the growth of gut bacteria, usually lactobacillus or bifidobacteria. Examples of prebiotics include lactulose, inulin, other oligosaccharides, lactulose, and resistant starch. And certain types of dietary fiber have also been shown to have a prebiotic effect. A symbiotic is a combination of a probiotic with a prebiotic, such that the prebiotic is used as a food source for the probiotic to selectively stimulate its growth. Other factors include the polyphenols in tea, cocoa, and wine. If bacteria have the enzymes that allow them to metabolize these polyphenols, they will have a selective growth advantage. In contrast, spices have been shown for centuries to inhibit the growth of different bacteria, and that's why they've often been used for food preservation. Well, I hope that I've shown you evidence to suggest that dietary components can influence both the numbers and types of microbes. However, this is a two-sided relationship, and the specific microbes that are present can also influence the response to dietary components. This slide shows a number of food components that can be metabolized by gut bacteria and the specific metabolites that are produced. Often these metabolites are more biologically active than the parent compound. Because of the short amount of time, I will just focus on the first two. So there is a strong relationship between dietary fiber and health. Dietary fibers are fermented by colonic bacteria to form short-chain fatty acids, including acetate, propionate, and butyrate. In fact, butyrate is the most widely studied and the preferred energy source of colonocytes. Production of short-chain fatty acids has been associated with health promotion and disease prevention. For example, the World Cancer Research Fund found that for every 10-gram intake in dietary fiber per day, there was a 10% decreased risk of developing colon cancer. There's also evidence to suggest that dietary fiber is associated with bacterial diversity. For example, in this study, the investigators found that when mice were on a high-fiber diet, they maintained their bacterial diversity through different generations. In contrast, if they were put on a low-fiber diet, they had reduced diversity, and this decrease in diversity was further reduced over subsequent generations. If the animals on a high-fiber diet were transferred to a high-fiber diet, there would be some recovery of the microbial diversity, but even less over subsequent generations. This means that the low-fiber diet leads to a loss of microbial diversity. Well, why is this important? As nutritionists, we know that a low-fiber diet is associated with an increased risk of many different chronic diseases, including diabetes, colon cancer, obesity. And we also know that a low-fiber diet is associated, we do know that these same diseases are associated with the decreased bacterial diversity. So this suggests that another mechanism whereby a low-fiber diet may be contributing to these diseases is through a decrease in bacterial diversity. In addition to affecting microbial diversity, dietary fiber can also protect the mucosal barrier. In this study, gnotobiotic mice, which is a model that's commonly used, these are mice that do not have any bacteria present. These mice were given a cocktail of 14 different human bacteria representing the 5 different phyla, and then were placed on 2 different diets, either a fiber-rich diet or a fiber-free diet, and then infected with an enteric pathogen. The mice fed the high-fiber diet had more fiber-degrading bacteria, a thick mucus lining, and are protected from the pathogen Citrobacter rodentia. In contrast, a fiber-deprived gut microbiota was found to promote expansion and activity of a specific colonic mucosa-degrading bacteria known as Akkermansia, and they are more susceptible to infection with the pathogen and developing colitis. Why this is important is that the availability of the potential food source influences the growth of different bacteria and can affect disease susceptibility. All of the mice had the exact same bacteria present, but the specific foods that they were consuming influenced which bacteria grew and influenced disease susceptibility. There is also evidence to suggest that diet and microbial metabolism can affect cardiovascular disease. Dietary carnitine, which is present in meat, and dietary phosphatidylcholine, which is present in cheese and eggs, can be metabolized by the gut bacteria to form trimethylamine. The trimethylamine goes to the liver where it's metabolized to trimethylamine oxide, and the formation of trimethylamine oxide, or TMAO, has been associated with increased risk of heart attack, stroke, and death in a number of human studies. Another important health problem in the United States is obesity. As you know, the obesity epidemic has been increasing over the last 20 years, and in fact, in 2018, all states in the country had more than a 25% incidence of obesity, being a BMI greater than 30. And as the cartoon says, don't step on it, it will make you cry. Similar to humans, some mice can eat more and grow more, but even more interesting, some can eat more and grow less. And that's actually germ-free mice. And the data supporting that is shown on the next slide. If you compare the germ-free animals that are shown in white to the conventional mice shown in black, you can see that the germ-free mice have 49% less body fat, despite the fact that they're eating 29% more calories than the conventional animals. However, if you take these germ-free animals and put them in the same cages as the conventional animals, they practice something called coprophagy, or they start eating each other's feces. And within 2 weeks, they'll have a similar body fat and chow consumption to the conventional animals, suggesting that the effects are mediated through the microbiota. There is also evidence from transplantation studies where you take those gnotobiotic or germ-free mice that I talked about earlier and give them the feces from different models of obesity, be it genetic obesity, the OB mouse, diet-induced obesity, where they're either fed a Western type diet or a fiber-rich diet, or humanized mice that are either obese or not. And in all those models, the germ-free animals that get the feces from the obese model gain significantly more body fat within a few weeks compared to those that get the feces from the lean model. And finally, there's evidence to suggest that obesity alters gut bacteria, again, in these different models. When you compare the lean to the obese animals, be it genetically induced obesity or diet-induced obesity, the lean animals have significantly less firmicutes, as shown in black, and significantly more Bacteroidetes, as shown in white. However, it is always good to know that the observations that you see in animals are also applicable to humans. In this study, 12 unrelated obese subjects were compared to 5 lean subjects. And as you can see, at baseline, the obese individuals had significantly more firmicutes and 95% less Bacteroidetes than the lean individuals. However, if they were put on a caloric or carbohydrate restricted diet for a year and allowed to lose up to 25% of their body weight, there was a significant increase in Bacteroidetes that actually correlated with the change in weight. I should note, however, in a number of meta-analyses, there was this relationship between firmicutes and Bacteroidetes were not seen at the population level. While there is a lot of evidence from animal studies that there is a relationship between diet and microbial composition, many of the studies have compared a high-fat diet with a chow diet, and the high-fat diet is usually purified diet, and there can be many other differences between these two diets. So in this study, they actually tried to separate out the effects of obesity versus diet on microbiome and glucose tolerance. Animals were fed one of 3 diets, either a chow diet that was rich in whole foods and had 15% of the diet as fiber, as either wheat, barley, soy, and maize. In contrast, the 2 other diets had purified ingredients. One was a refined low-fat diet, and the other was a refined high-fat diet. And both of these had 5 grams, 5% of the diet as fiber, and this was cellulose. However, the macronutrient composition of the chow and low-fat diet was similar. When the animals were looked at for fat mass and for glucose tolerance, the animals fed the purified high-fat diet had significantly more fat mass and a significantly less glucose tolerance. However, there were no differences than the animals fed the low-fat diet or the chow diet. In contrast, when the firmicutes to Bacteroidetes ratio was looked at in either the colon or fecal pellets. This ratio was significantly higher in the animals fed either of the purified diets compared to the animals fed the chow. So just to kind of put in summary that last slide, because there was a lot of information, what these data mean is that the high-fat diet induced changes in mouse gut microbiota composition due to diet, not obesity. Glucose intolerance and obesity in mice are linked to a high-fat diet, not to changes in the gut microbiota. Fecal fermentation or energy harvest decreased rather than increased by a high-fat diet. And that's pointed out because it was one of the mechanisms that was proposed early on to suggest how diet might be influencing microbial composition. But the most important fact from this study is that the choice of a controlled diet is vitally important in studies of microbiota composition. And I'm bringing this up to kind of segue into the presentation that Lex is going to give to better understand the data that she's going to present. So I hope I've convinced you that a dynamic relationship exists between the numbers and types of microbes, the food, energy consumption, and specific intake of dietary components, and that both of these can influence the specific microbial metabolites that are produced and thus disease risk. But the question comes, can we use this information to make personalized dietary recommendations? A key study was published about 5 years ago in which they found high interpersonal variability in the post-meal glucose observed in an 800-person cohort. What they found is if they looked at the glycemic response to a banana versus a cookie, some people would have a large increase with the banana, whereas other people would have a large increase with the cookie. And they wanted to understand, why is there so much variability? So they took information about their gut microbiome composition, blood tests, questionnaires, anthropometric, typical food intakes, and continuous glucose monitoring, and put this in the computer to develop algorithms to try and predict individual response. In a validation cohort of 100 participants, they found that the prediction is accurate and superior to common practice in this independent cohort. Moreover, they did a specific dietary intervention study in 26 participants to feed them foods that they predict would have a low glycemic response. And they did find that short-term personalized dietary interventions do successfully lower post-meal glucose. However, there's always the question of whether the results are reproducible. And in fact, last year, the study was reproduced in a population in the United States in which about 327 individuals were tested through the Mayo Clinic. And they did see similar effects, but not quite as strong. I am also pleased to let you know that the NIH approved 2 weeks ago in their Council of Councils $160 million for the study of nutrition for precision health powered by the All of Us research program, which will look at a cohort of about 10,000 individuals in the All of Us research program to better understand what is the relationship between diet, the microbiome, and health, and what are the features that we need to consider to be able to make personalized recommendations. So the take-home messages from my presentation is that microbiome research is an emerging area of science, and there are many research opportunities available. The microbiome is integral to human physiology, maintenance of health, and development in disease. In fact, there is a two-sided relationship between diet and the microbiome. While diet can influence the response to the microbiome can also influence the response to dietary components. There is an individualized response in how the microbiome will respond to diet and how diet will influence the microbiome. And finally, nutritionists, dietitians, and the food industry need to stay actively informed about advances in this field. The cartoon on the left says, I will have the garden salad, please. And the microbes are saying, we'll have the cheeseburger and fries. And it's important to realize that what you might want to eat is not exactly what your microbiome will metabolize. But even more important, that bacteria are your friends. Thank you, and I'll be happy to take questions later during the question session.
Great, thanks so much, Dr. Davis. I think in the interest of time, we'll go straight to Dr. Lobach. So Dr. Alexandra Lobach is a Senior Manager of Toxicology, Chemistry, and Regulatory Affairs in the Food and Nutrition Division at Intertek. And Intertek delivers expert scientific, toxicological, and regulatory consulting services. Dr. Lobach also has extensive experience working with low or no calorie sweeteners, also with probiotics, as well as other ingredients. So with that, Dr. Lobach, we will turn the platform over to you.
Well, thank you very much, Marie, for that introduction, and for Dr. Davis for the excellent first presentation. So I'm going to start with a brief background on low/no-calorie sweeteners, and to discuss why there's been such recent interest in the gut microbiota. Secondly, I'm going to look at some study design considerations when, as Dr. Davis went through, when you're studying the gut microbiome, but specifically when looking at how sweeteners impact the gut microbiota. And that will bring us to then looking at what the current literature reports and how we can apply these study design considerations to interpreting that data. And then lastly, I will dive into a few considerations for future studies and leave you with some key takeaways. So low/no-calorie sweeteners provide consumers with a sweet taste without the calories or carbohydrates associated with sugar and other caloric sweeteners. So they contribute few or no calories to the diet, as they can be used at very low levels due to their intense sweetness. So this allows them to be used in many reduced or low-calorie, sugar-free foods and beverages as an alternative to sucrose and other caloric sweeteners. And so several low/no-calorie sweeteners are permitted for use in the United States and globally, including examples such as acesulfame potassium, aspartame, saccharin, sucralose, and stevia glycosides. And these and other sweeteners have undergone extensive safety evaluations in several global jurisdictions, so beyond the US. And these include evaluations by scientific committees such as the Joint FAO-WHO Expert Committee on Food Additives, or JECFA. And this has resulted in the establishment of acceptable daily intakes for each sweetener. And so the safety databases that have been evaluated to establish these ADIs are comprised of standard toxicology data combined with clinical investigations and post-market data. So why this recent interest in sweeteners and their impact on the gut microbiota? So this really comes back to a high-profile study that was published in 2014 in Nature that concluded that artificial sweeteners induce glucose intolerance by altering the gut microbiota. And so this sparked a flurry of media attention. It made headlines in several mainstream media outlets and really reached the general consumer population. But this also captured the interest of a lot of research groups. And there was already research going on in this area, but it really sparked more investigation. So this really led to an increased publication in this field. So before getting into looking at the collection of literature and publications out there in this field, I'm going to take a step back and first go through some considerations when studying the gut microbiota and sweeteners, as this is really going to help us understand how to interpret the findings from these studies. So as I've been kind of going through this data set and looking at how to interpret it, I've realized that there are several questions that you have to ask when looking at the results in these studies. So the first question on the left, we have to be certain, are the observed effects on the microbiota directly attributable to the sweetener that's being tested? And so this relates to the study diet, which Dr. Davis spoke about in her presentation, and I'll just touch on a little bit as well as the form of the test article. As well, some questions arise with respect to the translatability of the findings in these studies to humans. So the second question is, are the study results dietarily relevant to humans? And so this relates to the dose of the sweetener tested in the study and the resulting exposure. And then another question is, are the study results biologically relevant to humans? And so this comes down to the study population. So I'm going to go into each of these points in a little bit more detail on the next slides. So first of all, looking at the study diet and whether the observed effects on the microbiota can be directly attributable to the sweetener that's being tested. So as Dr. Davis mentioned in her presentation, evidence indicates that habitual long-term diet and short-term dietary changes all contribute to the overall composition of the gut microbiota. And two primary mechanisms are hypothesized. First off, the microbial utilization of dietary substrates. So dietary components that are poorly digested in the stomach and small intestine, such as fiber, reach the colon and provide substrates for microbial fermentation. So those microbes that can ferment these substrates may have a growth advantage. The second hypothesis mechanism is the alteration of the microbial environment in the gut. So this is where dietary components change the physicochemical conditions of the gut, such as the pH or the presence of bile salts, and different microbial species differ in their tolerance to different environmental conditions. And so ultimately the take-home message here is that since the diet is considered to be a major contributing factor in determining the composition of the gut microbiota, it is therefore critical that studies looking at the effects on the gut microbiota strictly control for dietary intake. So the second item that can impact whether the observed effects are directly attributable to the tested sweeteners, the form of the test article. So when we talk about sweeteners, there are commercial formulations of these sweeteners, such as the tabletop sweeteners. These typically contain low amounts of the sweetener itself. So an example is Sucralose would be in very small amounts in its commercial formulation Splenda because it's a high intensity sweetener, but high levels of carbohydrate bulking agents are added to these commercial formulations, such as maltodextrin, to fill out the product. So studies should be conducted with the pure form of the sweetener so that we can be certain that the effects are due to the sweetener itself, or alternatively, proper control groups need to be included. As well, sweeteners should be investigated individually, not as a group. And this relates to their differences in chemical structure that lead to important differences in metabolic fate. And this ultimately impacts their potential to interact with the gut microbiota. So I've included 3 examples here on this slide. So if we look at saccharin first, this molecule is not metabolized, it's rapidly absorbed and excreted unchanged in the urine. We compare this to sucralose. Sucralose is also not metabolized, but it's poorly absorbed. And so it is excreted unchanged in the feces. So this provides it an opportunity as it passes through the GI tract to interact with the gut microbiota. Ribodioside A, which is a steviol glycoside, is hydrolyzed by the colonic microbiota to steviol, and that primary metabolite is absorbed and excreted in the urine. So we can see here these differences in these chemical structures and the differences in metabolic fate. So looking now at dose and exposure, and are the study results dietarily relevant to humans? So sweeteners that are permitted for use in food have been determined by regulatory agencies and scientific bodies to be safe for human consumption up to established ADIs. And so to ensure that a study has human dietary relevance, doses tested should not be greater than the ADI established for that sweetener. So in the table on this slide, I've included 5 example sweeteners here with their ADIs listed and their sweetness intensities related to sucrose. And so when we look at non-clinical studies, if the sweetener is mixed with food or drinking water, which is commonly done, it's therefore very important that consumption of both food or drinking water is monitored so that the actual exposure to the sweetener can be calculated. And bringing us to our last study consideration, looking at the study population or the subjects, and this relates to biological relevance. And so what I found through my review of the literature is that the majority of studies investigating the effects of sweeteners on the gut microbiota have been conducted in animals versus directly in humans. There also are several in vitro studies that have been published, but these studies are significantly limited in their biological relevance, and this relates to their limitations in being able to directly extrapolate the tested concentrations to human exposure levels. So it's very important to ask the question, is the gut microbiota present in the study population relevant to humans? And so a good example is if we look at mice. And so although the distal gut microbiota of mice and humans harbor the same bacterial phyla, most bacterial genera and species, so when we get more specific in their identity, found in mice are not present in humans. And when we look at this from a genetic perspective, only 4% of the bacterial genes in the human and mouse intestinal tract share considerable identity. So this just highlights that there are some difficulties when translating microbiome findings in animals to humans. And so a kind of a take-home message here is that a change in the gut microbiota of a non-clinical study population cannot automatically be considered synonymous with an adverse effect in humans. So now that we've looked at some of those study design considerations, I'm going to take us into a review of the scientific literature and what has been published to date on this topic. And so the aim here was to perform a quality appraisal of methodology in published scientific literature and investigate whether the available data supports the claim that low no-calorie sweeteners can alter the composition of the gut microbiota. So I carried out this study by doing a comprehensive literature search, and inclusion criteria were established. Articles were screened and included based on the 3 criteria listed here. So first off, all in vivo studies were included, in vitro studies were excluded. Studies that included obviously testing of a sweetener were included, even if there were 1 or more, but it had to have been administered orally, and there had to have been some sort of measure of microbial population present in the gut. So on this slide, I've included a table just overall summarizing the number of studies that were identified through this literature search. So 7 sweeteners have been identified in published studies. In the second column, you can see that the majority of studies have been conducted in non-clinical study populations, with a handful of studies having been done as well in clinical populations in the middle column. And so when you apply and go through these studies and look at whether there are any confounding factors, and so confounding factors here may include a dose in excess of the human ADI or a dose that's not reported, diet not being equivalent between groups or not controlled. So when we apply these presence of confounding factors, the number of studies that we can draw conclusions from become very limited. And so these are the number of studies listed in the number column. So for example, for asulfame potassium, although 4 studies were identified in the literature, only 1 study was identified to have no confounding factors. And in this study that was conducted in mice, in fact, no change was reported in the gut microbiota between the sweetener and control groups. Sucralose, there has been a lot of data published there with 13 studies. One was identified without confounding factors, and I'm going to go into that in a little bit more detail on the next slide. And Probiotics is another example where 2 studies have been identified. Actually, both didn't have any confounding factors. In the first study in mice, no change in the gut microbiota was found between the sweetener and control group. In the second study in rats, there was a change in the relative abundance of certain gut microbial populations between the 2 groups. So I'm going to go into a little bit more detail on sucralose so you can get a sense of what information was looked at in these studies. And so just quickly, sucralose is a non-nutritive sweetener that was approved by the US FDA in 1999 for use as a general purpose sweetener in human food. It's selectively chlorinated sucrose derivative, and it's approximately 600 times sweeter than sucrose. And as mentioned previously, its metabolism, it's not metabolized, it's poorly absorbed and excreted unchanged in the feces. The US FDA and JECVA have both established ADI values of 5 and 15 mg per kg body weight per day based on their evaluation of the safety data. And so if we look now at I think it was 10 non-clinical studies that have been published for sucralose, and this table and the next slide include the full details about the studies, changes that were reported in the gut microbiota, and then on the right-hand side, any study confounders that were identified. So if we look at the first study that was conducted in rats, this study was done with Splenda. And so from our discussions earlier on our study considerations, there was no control included in this study for sucralose alone. So we can't be sure if these changes in the gut microbiota were due to sucralose or perhaps the maltodextrin component of Splenda. As well, there were significant differences in body weight reported in this study, and food consumption was not reported. So this could mean that food consumption was different between the different groups since body weight was different. So we can't be certain that these reported changes in the gut microbiota are not due to these differences in the diet. So if you go through the study confounders on the left, you can see that some of these other studies administered doses in excess of the ADI. As well, there were several conference abstracts that were identified, which definitely often do not have enough details to draw conclusions from. And moving on to the next slide, just summing up the other studies that were looked at. As you can see, there were 4 others here that had study confounders, with one study that I did mention being identified that did not have any study confounders. This study was conducted in mice, doses were below the ADI, diet was controlled for an equivalent between control and treated groups. And one change in the gut microbiota was reported, and this was a decrease in a fecal Clostridium factor, and it was reported to be dose dependent. However, there is no evidence that this change in the murine gut microbiome is translatable to an adverse effect in humans. So what if we look at the clinical data on sucralose? So 3 clinical studies have been published looking at sucralose and its impact on the gut microbiota. 2 studies, the first 2 in this table, are conference abstracts only, so there are a lot of limitations here with respect to interpreting those results with confidence. But if we look at the bottom study in gray, by Thompson et al. In 2019, this study was conducted in healthy adult men, and there were the reports in the gut microbiota, but interestingly, there weren't any changes that were reported between the sucralose and control groups. So first off, the relevant abundance of several phyla were reported to remain stable between the 2 groups, and a principal component analysis was conducted for each subject compared to their baseline, and there was a close similarity reported for both control and sucralose groups. There was one study confounder identified in that the diet was not standardized between the control and sweetener groups. However, it doesn't appear that that would impact the reports that were found with respect to the gut microbiota. So overall, the data on sucralose don't tend to suggest that there is an impact in humans. So moving on now to some considerations for future research. So in addition to those study considerations that I already discussed, it does appear that properly designed clinical studies are the most relevant model to directly study if there exists an association between sweetener consumption and changes in the gut microbiota. And so these studies can be conducted in a safe manner, as doses up to the ADI are considered safe for human consumption. And it's expected that any observed changes in the gut microbiota would be transient. So if sweetener consumption was stopped, those changes should revert. As well, Dr. Davis talked about this point a little bit. So the use of animal models that are more biologically relevant to humans with respect to the gut microbiota. And so this relates to those rodent fecal transplant models. So where germ-free mice receive a fecal transplant obtained from humans, allowing them to establish a gut microbial community that is reflective of humans. So that sums up my presentation, and I'm just going to leave you with a few key takeaways. So as we discussed, several scientific studies have been recently published that evaluate sweeteners and the gut microbiota, but appropriate study design is critical to ensure that results from such studies are biologically relevant and not confounded. And so overall, the current published data collectively support that there is no clear evidence suggesting that low-no-calorie sweeteners adversely impact the gut microbiota when consumed by humans at approved levels in the diet.
Great, thank you, Lex. I'm just going to share my screen here. Okay, so we have a couple of questions and a little bit of time. So one question for I guess for you, Dr. Lobach, is about other sweeteners. So stevia more generally, as opposed to rhodioxide, and monk fruit sweeteners, those are pretty popular. Can you tell us anything about the effect of those on the microbiome? Have they been well studied?
Well, so as I mentioned, in my search of the literature, only identified 2 studies on steviol glycoside. So the search terms did include steviol glycoside, stevioside, so beyond rhodioxide. And in fact, rhodioxide A was the only sweetener, steviol glycoside, that came back with search results. So those 2 studies are the only ones that I'm aware of in the published literature to date on steviol glycosides. Monk fruit extract was included in the search results, and I didn't find any studies.
Okay, great. And I think this is a really good question. So there have been a lot of presentations that express concerns about sweeteners in the gut microbiome, just like the articles that you had showed in those titles. So why are there these differing perspectives out there, or people hear different things when they receive presentations on this topic? Can you comment on that?
Well, I think, as I mentioned, it's really important to look beyond the headlines that these articles or these, take-home messages that the articles may state. And we really need to look at it from a scientific and critical perspective to ensure that the results that are reported are directly attributable to these sweeteners, and to take it one step further to really understand how and what this means for humans as we consume these sweeteners through the diet.
And I'd like to add one other comment, is there can be small changes in microbial composition that aren't functionally important. And ultimately, it's the biologic function rather than actually who's there, but what they're doing. That's a lot more important. And there is a lot of redundancy.
Great, thank you. So there's one question here about the research that needs to be done on this topic. So we're obviously, this is kind of an early science, and there is a lot of research out there, and we're continuing to evaluate it. But what needs to be done to really understand the role of sweeteners on the gut microbiome? Do we have adequate methods at this time to give us a conclusive result, a conclusive answer?
Well, that's a really good question. I think more studies should be done, and I think that we should look at doing, as I think in my second last slide, conducting these studies in populations that are relevant. In terms of the analytics, the techniques to evaluate the gut microbiome have improved immensely over the last decade. And so the information that can be obtained from the gut microbiome is much more than we used to be able to obtain. But I think we also need to, as Dr. Davis mentioned, understand what those changes mean and truly understand whether those changes actually mean anything, and let alone if it's an adverse change or not. And so I think that's where a lot of understanding needs to be had.
Yep. Okay, super. So there's one question here about the ADI, and in one of your slides, Dr. Lobach, you showed that the sucralose ADI, for example, was quite different between the US and JECFA. And so the question is, does this reflect the different authorities' perceptions of safety?
Well, that's a very good question, and good to catch that from my slide. So yes, in the case of sucralose, the FDA and JECFA derive different ADI values. And so this just boils down to the fact that they evaluated many safety studies in their evaluation and derivation of the ADI for sucralose. And so I believe that the agencies ended up deriving different no observed adverse effect levels based on slight differences in interpretation of the safety data. The ADI is not, in fact, that different between the two, and so there is no difference really in their safety interpretation. It just comes down to difference in scientific interpretation.
Thank you. And I think we'll just take one more. And Dr. Lobach, this is another one for you. So you mentioned that sweeteners should be studied individually. So the question is, does this mean that any findings are only applicable to the particular sweetener that was studied in the scientific paper? And then if that's the case, how should we interpret headlines that use collective terms like you had shown, such as artificial sweeteners do X?
Well, that's a very good question. And so, yes, sweeteners should be looked at individually. And so this relates to the fact that they're each metabolite, they're different molecules when you think of it from a chemistry perspective, and they're metabolized in different ways. But a good example, if you look at steviol glycosides, they're all metabolized to the same core steviol backbone. So that would be one case where a investigation on ribodioside A could apply in its conclusion to the entire class of steviol glycosides. But when we look at other sweeteners, they should all be considered individually. And the second part of your question
was about the, if we see a headline or any kind of statement about artificial sweeteners as a group do something, how should that be interpreted?
Yeah, so when you see a headline like that, I think it's very important to go right back to that published research article and find out what sweeteners were tested. Was more than one sweetener tested? Did they see the same results in each sweetener? So I would say you really have to be careful interpreting those blanket headlines.
Great, thank you. Well, we've reached the top of the hour. We do have more questions, but we'll think about developing other content to look at those. So that brings us to the close, and we wanted to give a huge thank you again to Drs. Davis and Lobach for sharing your insights with us today. A reminder to everybody that you'll shortly receive a very brief survey, so complete that to receive your CEU certificate. And if you're interested in more information about the science around low-calorie sweeteners, we do have a session at FNCE 2021 on October 20th. So we hope that you will join us there. So thanks everyone again, and we hope you have a great rest of your day.